This conference will now be recorded.
Yes, Dr. Anand, we can see. Good evening, everyone. I am Rajni Jain. On behalf of agricultureinformation.com, I'd like to welcome you all to this online meeting. Please take a moment to review the guidelines for this online meeting posted in the chat window. I now request the moderator, Mr. Lakshmipati, to introduce the speaker and start the meeting. Thank you.
Thank you, Ms. Rajini. Thank you, Ms. Rajini. Good evening, everyone. I am Lakshmipati. I'll be moderating this meeting. Agricultureinformation.com likes to welcome to this meeting Dr. Anandkumar Naorem, Head in Charge and Scientist of Soil Sciences at ICAR-Central Arid Zone Research Institute, RRS-Bhuj, Gujarat. Dr. Anand Kumar will be speaking on the topic, why farmers should test their soil. I now request the speaker to begin.
Okay, I'd like to get a confirmation whether my screen is visible now.
Yes, your audio is quite clear.
Okay.
The screen is also okay. Please go ahead. Pardon? Yes, your presentation is also visible.
Okay. Thank you. Okay, I would like to start to all the members present today. A very good evening from Kajri. I am Dr. Anandkumar Naorem, and today I'll be delivering a very simple topic, yet very important. And I guess most of you, although you must have an idea of soil sampling, must have a series of several questions in your mind. Although this will be put up as a recording in your website, I guess that most of the things that deals with the soil sampling, I hope I can answer all the queries of the people through these slides. But because of my tight schedule, I just finished preparing the slides before 10 minutes of the lecture. So if there is any confusion, please let me know. And if this recording comes up in your website, if you have any further questions or queries, then you can contact me. So let's start some soil sampling then. You all care of your own people. We do take care of our parents, our family, our friends, etc. But we sometimes fail to care the mother that gives the food for us. Yes, the soil. So the soil system is a very unique natural resource that supports our life. And I don't need to emphasize how the soil supports life. I think everybody knows about it. But I would like to highlight some additional things about the soil system. You know that plants grow on the soil, therefore soil gives food to us. Moreover, soil contains millions of living organisms, millions and millions of living organisms. And without these organisms, I cannot call soil as soil. It will be a dirt. So these microorganisms are used for several purposes, including your medicines, I mean your pills, your tablets, everything. But due to our human activities, our anthropogenic activities, lots of toxic chemicals and pollutants are loaded into the soil system. And we are getting these contaminants in our body through the food chain or food system. So you might be knowing why so many causes of cancer, liver diseases, kidney dysfunctions, and so on, despite of taking very good food in the long run. So therefore, soil is the ultimate foundation of life. So this beautiful natural resource is unfortunately degrading day by day. But what are the reasons? Climate change and human activities, obviously. So if we see these maps, I hope that these maps are visible in your screen. This, if you see in these maps, it shows in India, the circle part is India. So the soil loss or the soil erosion is at a very high rate, and many of the places are in degraded states or in very highly degraded states, meaning they are unfit for cultivation of crops. So this raises a huge concern for our sons and daughters and their future generation. So what we can do as a farmer, what we can do as a citizen, what we can do as a a person who has been getting the food from the soil. As a farmer, your soil is your best friend. So you feed and take care of her, she will give you what you actually want. Since the soil doesn't know how to speak, we need to decode what the soil wants. So you send the soil samples for testing. So if you ask me why to test, number one, to understand what is the health of your soil. If you need to understand how much nutrients can the soil give to the plants and how much is accessible to the plant roots for absorption. And number two is if you know how many of this nutrients your soil needs, you can apply that amount of fertilizer only, no deficit, no extra, thus saving your inputs, your money, as well as reducing the environmental pollution. So you can also know the health of the soil through deficiency symptoms in plants. These are beautiful pictures that shows how a nutrient deficiency can cause specific symptoms. If you see on the left hand side, this is a phosphorus deficiency in chickpea. This is beautiful. This picture is one of the best, photo awarded in IPNI. You can see the purplish color in the leaves on the lower parts of the leaves, which is phosphorus deficiency. In the middle, we can see the potassium deficiency in soybean, where there is a necrotic leaf at the upper layer of the the plant, and there is a potassium deficiency in wheat. Although these pictures are very beautiful, this depicts how your soil is deficient. What is the health of your soil? You can see the boron deficiency in sweet potato. These potatoes are wrinkled, and what is the use of this sweet potato when they are completely wrinkled? They don't have a market value, and you cannot consume it. And there's an intervennal chlorosis in the mango due to the magnesium deficiency. So all this element has their own way of manifestation their deficiency in through the deficiency symptoms. So each element, they have a different deficiency symptoms, and each plant also they have a different deficiency symptoms, you can guess. But there are three problems with deficiency symptoms. Number one, this deficiency symptoms mostly occur after the damage has already been done. Number two, there is a system called hidden hunger, in which the plants do not show any symptoms, although there is a deficiency of a nutrient in the plant, but affects its productivity. It often happens in the case of potassium also. A deficiency symptom of a nutrient can very much resemble with the pest infestation, and it might confuse the farmer whether he or she should go for a pesticide or whether he should go for a fertilizer application. So before the start sampling, let us know what kind of errors we might get during the whole process of sampling. The point is why the samples are collected. This sample means a small part of the soil that can represent what the soil actually is. So if we take a wrong sample, then we will give you wrong data, then wrong action. So let us see what kind of errors are associated during each level. The first error is during the sampling, maybe due to the wrong method or due to the selection of the abnormal spots, which I will discuss later. The second error is subsampling, where the soil has a different layers of soil profile. So when you take from each depth, maybe there is a mixing of the soil from each profile, and it might give a wrong data. And the third is analysis error, which might be due to the manpower, which might be due to the technical error, or which might be due to the instrumentation that you have been using during the analysis. So what kind of tools we need to use for soil sampling? So this is completely, I'm trying to avoid most of the scientific jargons, and I'm trying to avoid most of the scientific basis of soil sampling. Most of the information which I'll be delivering through my lecture will be very, very basic and important for the farmers and farming community, or not only for the farming community, but those people who love to go for farming in near future as well. So these are the different kind of tools that you can use for soil sampling. We mostly use in India, we mostly use spade. I think spade is more than enough for common collection of the soil sample from the surface layer. It is used in soft and moist soil, and there are other type of soil sampling tools, also screw type agar, which can be used in hard and dry soil, which are very which have a large portion of stone and gravels. You just push the auger into the soil and just drag it out, and within this ridges you will get the soil samples. And you have a tube agar, the soft and the moist soil, and when it is free from gravels, it's better to use tube agar. And you have also a Dutch auger, which can be used in moist and wet soils, but only for the fine texture kind of soil. But most of the people, I think, which have interacted, they have used khurpi or spade in most of the cases. So let's start some soil sampling, I guess. So this is your own field. Due to a little bit of my short time, I could not represent graphically very well, but I guess the point which I want to deliver here can be easily visualized. So your own field, right? So the elevation, how much it is depressed, kaha depression hai, kaha abnormal spots hai, kaha calcium carbonate deposition ho raha hai, where the water accumulates more, where the water doesn't accumulate at all. So you know it. But if you don't know, okay, I mean, you make a walk around a field and you see the variability in the field. Does the soil color changes within the field? Do you feel the texture different in one part and another part of the field? You just pick up the soil and you just feel the texture of the soil. Maybe you will be able to see the difference in the texture from the left side of the field to the right side of the field. Then try to mark the spots. Try to collect samples from same type of areas. For example, this on the left hand side of the field, you can see the green ones are same, brown ones are same, and orange ones are same. So from this field you have 3 different types of samples. And you also try to identify the abnormal spots. Abnormal spots means where it is different from the main general characteristic of your own field. Maybe water accumulates more, salt depression occurs, that's all. And another one is avoid boundaries of the field and within the field. So you try to avoid the boundaries of the field and within the boundaries within the field. Under each boundary, you make random spots and collect the soil samples from these spots. You try to make random spots in such a way that it reaches most part of the field except boundaries and abnormal spots. So this is not for the scientists. This method is completely for the people who have been giving soil samples to the laboratories for their own testing for their own field. And those scientists or those students who are joining here who will be seeing this video afterwards, you cannot follow this procedure for your experimental analysis. There is a specific procedure. I guess you will be finding this in internet as well as in good books. And if you are not able to clear for it, that you can contact me. But this is for the general soil sampling. Then you try to make these random spots in such a way that it reaches most part of the field except boundaries and abnormal spots. Okay. Then you try to make a V notch while taking a soil layer. Make a V from each side, make a cut from each side so that it takes the shape of a V. Then dig out a layer from one side of the V, like you cut a cake from a vessel. Very easy. It's called a V notch. So if you ask me how deep we should take the soil sample, see, most of the plant's roots stay within 30 centimeter of the soil depth. So if you have a common crop and a seasonal crop, then up to 15 centimeter to 30 centimeter is really fine. But if you have a tree crop like mango or a big tree where the roots go beyond 30 centimeter, then take up to 60 centimeter if possible. With each soil profile, the soil properties might be different. The quaterings method, what we can do is that this is one of the very basic method, and this is very important in case of soil sampling. You collected a sample, abhi aapke paas samples hai, a huge amount of the sample you have, then you mix it properly, then you divide into 4 parts like this, 1 2 3 4. Then you discard the opposite sections like yellow, you just take it out and you keep it this sections. And you take this green sections, then you mix it again, you take out the 2 other sections till you get like this around half kg of the soil, like 500 gram. It depends, 500 gram is not fixed, it depends on how many parameters you will be doing it. So before you submit the soil samples to the laboratory, you will just get an idea from the manpower of the laboratory that how much gram of this soil actually you have to submit for this soil analysis. Yeah, so some questions are from where the soil samples should not be taken. Number 1, plowed soil, जहाँ पे अभी tillage ही हुआ है, अभी ploughing ही हुआ है, तो we should not take from a very fresh plowed soil because the soil is already disturbed and mixed, and there is no point of getting the idea from a recently fertilized field because already वहाँ पे fertilizers लगा हुआ है. Then unusual spots like calcium carbonate depositions ho ya stone, bada stones hai, I don't think we can easily avoid those spots. The depressed area where there's water used to accumulate, not only the water but the nutrient tries to accumulate, so we try to avoid this kind of area. The manure heap where you make a pile of a manure, because manure itself is very rich in organic matter and nutrients, so if you take the soil sample near and around the manure heap, that it might over calculate the amount of nutrients that you have in the soil. So as I have told you, that wrong samples means wrong information and wrong action. And contaminated area, so if it is contaminated, then I think we should better try to avoid it. And near border area. So you have collected the soil sample, so what you're going to do, you're going to bring the soil sample from your field to the lab. You collect the soil sample, put it in a plastic bag, and you label with the name of the farmer, with yourself, the name of the village, the GPS coordinates. GPS coordinates is nothing but latitude and longitude, the area to know where it is taken. And I think most of the people around here, they are mostly acquainted with phones, mobile phones. So what you can do is that you just can download any app of GPS coordinates. There are lots and lots of apps in Android as well as in iOS. You just have to open that app and you just see, okay, this is the area where I'm taking the soil sample, so this is the coordinate. You just have to write that coordinate and submit to the laboratory. What it does is that it is not actually useful for you, it is actually useful for the whole country, because the laboratory, when they have a compilation of the GPS coordinates and the data or the soil data, they can identify what is the spatial distribution, how the soil is distributed in our country, in our district, in our block, and they maybe identify some of the spots which is really need immediate attention. So this is really important. And if you want to have a quick contact with the laboratory people, then you can also jot down a phone number in the label. Another thing is that you have to make sure that there are no not soil materials. Not soil materials means plant roots, plastic, gravels, जो soily नहीं है. So when you collect the soil sample, you did the quartering system, you divide it into 4 parts, you have a 500 gram of the soil sample. So I think it will take only 5 minutes to sort it out. Just spread it out on a newspaper or a clean paper, and you just see, okay, what are plastics hai kya, छोटे छोटे plastics hai kya, plant roots hai kya, या stones hai kya, because this will really interfere in the soil analysis, especially these plant roots, because plant roots, they are having some part of the nutrients, they are having some part of organic matter, so it might interfere completely with the soil analysis as well. You can further help, but this is not at all compulsory. You can further help if you have this facility in your home. You can spread the soil sample over a flat surface and air dry it under the shed at room temperature. And if you have a sheaf of 2 mm mesh size, then you can save it for getting a uniform soil particles. I would like to highlight this: when you give to a soil analysis to any laboratory, please make sure and tell them, okay, do you sieve the soil before analysis? Let them know that it is very important to sieve the soil, because some people out of lethargy, they don't sieve the soil and it creates the wrong data. You need to have a uniform soil particles in order to go for analysis. So either you can do, or you can make sure that the people in the technical people in the soil laboratory, they do as well. So where should I send my soil samples for testing? This is one of the very common questions that people ask. Any recognized soil laboratories? I think in India, ICAR has many institutes, and you can send to any institutes, nearby institutes, KVKs. Then there are some of the private institutes as well that comes up with the idea of doing the soil laboratory analysis. But I don't know about the pricing. It might be a little bit higher in private sector, but it totally depends on you. But you have to make sure that they have enough analytical facilities. And in my opinion, I think it's better to avoid those laboratories that uses the multi-nutrient soil kit. Multi-nutrient soil kit is nothing like when you go to a laboratory, some people may come up and say, हमारे पास एक machine है, बड़ा सा machine है, आपको soil sample डालते ही वही बताएगा कि वो how many nutrients are there, how many nitrogen, how many phosphorus, how many potassium. The answer to this question is, unfortunately, there is not a single instrument that can absolutely give a very good idea of all these nutrients, the soil quality in one go. So whatever it is there available, they have a very less accuracy. If you don't have this facility, then you can do it, but in my opinion, we should try to avoid those multi-nutrient soil kit. In our laboratory, we have enough manpower and enough instrumentation that deals with each parameters, each nutrients for and they do it analysis separately. We never use a multi-nutrient soil kit. And सबसे important बात है कि you send the soil samples to where you can get the interpretations and scientific advisory. आपने soil sample submit कर लिया, you got the data from the laboratory. Then after that, what? There is no use. If you got the data in order to know how it will be helpful for you. So you need interpretations and scientific advisory. So the interpretations will be given by whom? The people from the laboratory. So the laboratory people or the scientist who is associated with it must be able to make you understand that अभी soil में कितना nutrients कमियां है, क्या problem है, कैसे ठीक कर सकते हैं. Okay, आपका electrical conductivity बहुत ज़्यादा है, then how we can reclaim that saline soil, कैसे करना है. You should be able to give you recommendations, not like ये इसका 2% है, इसका 5% है, इसका 6% है. These are just a number. We as a farmer, as a gardener, as a person who wants to do gardening or farming in near future, you are mostly you want the the work that should be done, not the data. So please get acquainted with a soil laboratory that can also provide this scientific advisory to you as well. Some of the questions in your mind that might comes up while this whole presentation. The first question may be best time of sampling the soil. If I have to say, the best time of sampling the soil is autumn after Kharif. So when you finish the Kharif season and when you go for the autumn season, that's really good. But if I have to say, okay, autumn to miss kar diya, kaise karenge? You can take any time of the year, being provided that you do with the correct method, and being provided that you get the sample in such a way that the sample represents the actual soil that you have in your farm. And how many in how much intervals of soil sampling we have to do it, like we have to do every month or every 6 months. It's alternate year, so you can do it alternate year. If you are unable to do alternate year, you can go for two after 2 years, but it's best and it's recommended to go for alternate years. And do we have to dry our soils? Absolutely, you have to dry your soils. Air dry, not with fan or with oven or under sunlight. You should not do it. Only under the shade, in the room temperature, not with fan or anything. It should be dried. But for the biological analysis, suppose for example, there were a lot of entrepreneurs who really wanted to know how many bacteria, viruses are there in my soil. I just want to know about it. So for that, we need a fresh wet soil. And for the nitrate or extensible iron also, we need a soil which is not at all air dry. If you ask me, okay, my crop is already being in the growth phase, in the standing phase, how can I collect the soil sample now? Do I have to wait for harvesting of the crop? No, you don't need to. You can collect the soil samples between the areas between the rows of the standing crop. Be sure not to harm the roots of the crop, and be sure you collect the soil sample as per the methods and protocols that I have mentioned in my earlier slide. So this is the end of my presentation. I guess that this has given a very simple perspective of soil sampling, and I try to avoid most of the scientific way of soil sampling because this is very important, but some of the parts it might not be understood, as well as it might not have been, you might not have crossed previously. So I try to incorporate as much as possible. So if you have any questions, you can ask. Otherwise, you can further contact me after seeing the video of this recording. Thank you.
Thank you, Dr. Anand Kumar, for an interesting presentation. I now request the participants to ask their questions. We'll also be posting the speaker's contact details in the chat window. Now, over to the participants for questions, if there are any. Please indicate your willingness to ask questions by putting on your mic, please. Any questions? Mr. Pamati? Yes, Mr. Mati.
Yes. Hello. Hello.
Mr. Mati.
I am
Yeah, no,
audible.
please.
Hi.
Hello.
Yes, Mr. Mati, please go ahead.
So okay. So thank you, Dr. Anand Kumar. It's a very nice presentation. So you said at the last end, you told about some interpretation. So if you get a, can you explain with some sample data and how you can interpret our typical interpretation for a typical sample?
Yes. This is a very good question. I think I have missed out in my slides. This is one of the very good questions. When you when you give this first sample to a laboratory, after analysis, they will be giving a sheet of paper, isn't it? So they will be giving a nitrogen this much percentage, phosphorus this much percentage, the potassium this much percentage, organic carbon this much percentage. So what you can do is that you can go to the technical person, or you can either go to the scientist associated in that laboratory and ask him that, okay, what I need to do, is my nitrogen more in my soil or is it less in my soil? Interpretations means, for example, nitrogen, there is a limit of nitrogen, there is a specific limit of nitrogen in our soil that should be. So if your data comes beyond that limit, then you need to either put nitrogen, or either you have to do something that in order to take out as much as the excessive nitrogen. So that's called interpretation. And another thing is that for fertilizer calculation, for example, your boron is very less in your soil. It is less than 10 ppm in your soil. Then you will be you are telling to the scientist, oh, I have been applying urea, I have been applying DAP, I have been applying organic manure for so long time, but it's not at all working. Why is it? Then boron may be a deficient micronutrient in your soil. So from the analysis, you see that boron is below 10 ppm. So you tell the scientist to calculate and tell you how much of borax you have to give to the soil. And if you are unable to get a scientific advisory, then there are a lot of applications in the mobile phones. You just have to download fertilizer calculation app. Then you just put, okay, my boron is 5 ppm, then I'm going to put wheat crop or rice crop. Then they will tell you how many of borax you have to apply, when you have to apply, Then is there any alternative fertilizer other than borax if you are an organic farmer? So that kind of thing, these are the interpretations that you have to collect from the scientific advisory or either from the application in the precision agriculture. I hope
One
I
more
think.
question. Yes, I can understand. Say, you told about some application. Can you name something which is available, or are you can yourself do it in your as an institute, isn't it, for the benefit of the farmer, the government
Yeah.
funding?
Yeah, for me, I think I'm a little bit old school, so I have been well informed that there are a lot of apps in the application, in the mobile phones. I have seen that also. So if you type fertilizer calculator in Android, I think there are some apps, but I never use that apps, although they are quite feasible and they are quite accurate. I try whenever a farmer comes to me and shows their their data, I try to calculate myself in the paper. I love to calculate and let him understand, okay, how much of borax I have to give them and everything. So that's I used to do. So that's
So,
why I prefer that you go to a scientific advisory to get that interpretation.
okay. One last question, sir. It's whether the soil sample result will give us a pH or EC or the EC conductivity. How can you
uh,
say when you say that to pH, you can say acidic or alkaline soil, so that we can
hmm.
make a corrective measure, gypsum or something like that we can add to correct it. But the EC, what will indicate? Nutrition
Okay.
uptake.
Okay. pH and EC is quite different. pH is like whether it is acidic or whether it is it doesn't, yeah, alkaline. But pH doesn't actually say the alkalinity of it. pH just gives an idea, okay, whether it is soil is acidic or alkaline. EC means how much of salt are there in your soil. For example, although it is alkaline, there might be less salt, there might be more salt, and there might be different kind of salt. If it is saline salt, and there are two kind of salts also, it may be saline or it may be alkaline. So according to it, you have to give different kind of approaches. So with pH only, you won't be able to know whether it is alkaline or saline, although you might be getting a little bit of overview. But with EC, you will be able to know that how much of these salts are accumulated in your soil, which is really, really important. So along with the EC, along with the pH, then it's very recommended to get a analysis of EC.
Okay. Okay. Say, well, regarding the EC, that we are using drip irrigation with the liquid fertilizer to the root zone. That the EC have an effect on that intake uptake of this nutrition to the root zone.
Yes, yes. If it is very high EC, see, it is very simple. The EC means salt. The salt means sodium. For example, sodium. Sodium loves water. They love water, so they try to cover themselves with water. So if there are a lot of sodium in your field, in your soil, whatever water it comes to your soil, they will try to cover it. They will try to use it to cover them. So they won't give to the plant roots. So you'll be giving a lot of water, but the water will be going for making a blanket around the sodium, but it won't go to the plant roots. So you might be seeing some of the farmers, I have been applying a lot of water, I've been applying drip, I've been applying flood irrigation, but I don't know, this plants are always wilted. They are not giving any, they are not having any freshness in their leaves. Sometimes it's because of the sodium. There are a lot of sodium. If it is a high EC, then the sodium tends to accumulate lots of the water around its body and make it unavailable to the plant roots.
Okay, thank you. Thank you. I cleared about pH and the EC. Okay, thank you.
Yeah, thank you, Mr. Mati. Any other questions you have? I guess there are a lot of people who I know personally, so I think they won't. I think they are.
Maybe student or associates with your institute.
Yeah, because they won't ask because they know.
Good evening, sir.
Yeah, good evening, Mr. Kuldev.
Yes, sir, I want to ask something. That we are actually very basic level. We are at the basic level. When we go for initial grouping the seeds, we used to have two numbers of DAPs, 50 kg, 4 on zinc before we
I'm sorry, but I'm unable to hear your voice. I mean, it's quite noisy. Could you please mark that?
He can put in a chat window his question.
Mr. Zala,
Yeah,
there
you
is
can
a
either
lot of
put it.
background noise from your end.
Hello.
The background is little bit noisy. Yeah,
Hello.
yeah, yeah.
Yeah. Pardon, sir. Actually, I want to ask this. We are from we used to have two numbers of BP days when we are working initially in the
Mr.
starting
Zala,
of career or
I'm so
Mr.
sorry.
Zala, there is a lot of background noise from your end. Post your question in the chat window. Yeah. So other participants, would you like to ask any questions?
Mr. Kuldeep Singh,
Yes.
any questions, please? Have you posted your question in the chat window, please? No, he's not able to do that, and he is not available also, looks like, and he's not responding to me also.
No
Anyway,
problem.
we will wait for others.
Contact details are always shared, I think, so you can contact me later on.
Okay, thank you. That will be better. I don't see any questions in the chat window either. Okay, then thank you very much. And it's time to thank the speaker, Dr. Anandkumar Naorem, on behalf of Agriculture. I now request the organizer of the meeting, Ms. Rajini Jain, to please close the meeting. Thank you.
Okay, I think, sorry, Mr. Lakshmipati, I think Mr. Kuldev has put any chat box, I think, question.
No, he is yet to post his question.
Okay,
Not
he
able
has
to.
sent, yeah, he has sent to me, I think. He asked how can we locate ICA laboratory at our location?
Recite the question, then we can answer, please. So, for the sake of other
Yeah.
participants.
So Mr. Kuldeep Singh Jala, he has asked, how can we locate the ICA laboratory at our location? So I guess Mr. Kuldeep Singh Jala is located which part of India? Is it in Gujarat?
Yes, in Gujarat, near Morbi.
Near Morbi. Okay. In Gujarat, we have a lot of ICA institutions. We do have in Bhuj, as I am in now. Then we do have in Bachau, not Bachau, this one, Junagadh. We have. Then uh, there is one soil salinity institute, Baruch. They have an institute. So I think you can send the samples to them, or plus if you have any KVKs around you, then you can go for that laboratory as well. No need for the ICN institutes for any state government, or you can also target those academic universities, any state agricultural universities. They also do the same. So they also provide services like soil analysis.
Okay, sir. Thank
And
you,
if you
sir.
type,
Also,
if you type ICN,
sir,
yeah.
we want to ask something more.
Yeah.
Sir, we are very basic level at farming level. We used to post simply two numbers of DAP, NPK, and somewhat some other combination like 1232 and all that thing at initial while sowing the seeds.
Yeah.
So whether it is a correct method or it's a rough calculation given by our traditions.
Yeah. The traditional mode of applying fertilizers, they are wrong in so many levels. It's just the amount actually, because we never measure. We just get a bag, then we see, okay, itna bag lagana hai, itna meg dalna hai, itna hi handful dalna hai. We have an idea of it. We just apply it, but we never know what the soil actually needed. The soil might be having a lot of nitrogen, and you are just putting urea thinking that, okay, if I put this urea, then it will be working in near future. So this urea will be exposed to volatilization. It might go into the atmosphere, or it might be drained out with the water, so it increases your input cost. So that's why it is really important to get the soil test done in order to understand how much of soil nutrients you have to give, because this broadcast method and before sowing you just put it how much of it, and आप pit भी बनाओगे तो थोड़ा थोड़ा डाल देते हो. So you must be thinking that okay, I'm putting little bit, but if you calculate by your whole big hectare, it might cost a lot. So this has a huge problem. So in order to increase your productivity at the same time to decrease your input cost and to think about the environmental pollution, then I guess that it's really recommended to go for a soil test, and it's very affordable, I guess, if you do from a very recognized soil laboratory like from our institute.
So we can actually contact you when we are in the Bhuj location, that some many problems are existing because it is continuing. These traditions are continuing from last 20 to 30 years.
No problem, no problem. You can come down to Bhuj with your soil sample, and we have enough technical member as well as I'm also there to guide you whatever it is, and we can discuss afterwards what problems and what issues are there in your soil.
Okay, thank you. Anything more, Mr. Kuldeep Singh? Okay, I have one more, just one more question before Dr. Anandkumar. Maheshwari has posted a question: which way is easy to collect soil?
Okay, which way is easy? I guess he might be asking which method is very easy to collect soil. The V method, which I have told you. Yeah, the V method, which I've shown in the slide. This is a soil. You just make a cut from this side, then you just make an opposite incline this side. Then it will make a V. Like you just put a V in the soil surface. Then from one side of the V, you just scrap the layer. You just scrap it because the layers will be exposed to you. So that's called V-notch. Some people, they do very reluctantly, they just put the spade in the soil and dig it out the first layer and just put in the soil. It's not the actual process. You just make a V in the soil, then from each side, one of the sides, you can make a thin layer, like you used to cut a cake in your birthday. It's very simple. So this is the easiest way to collect the soil sample. But if you want to collect from the below levels, then I recommend you to go for augers, because in augers you can maintain the depths and you can go inside the soil very easily, the soil system very easily. Otherwise, PET, I think it's only for the surface layer. So I hope I can answer this. I have already answered this question, I guess. Any other, please?
Thank you. Thank you, Dr. Anandkumar. I it's time to close. Ms. Rajini, please you can close the meeting.
Thank you, Mr. Lakshmipati. I'd like to thank the speaker, Dr. Anandkumar, for taking the time to talk with us. I also like to thank all the participants for their time. The meeting will now be closed.
Thank you so much.
You're welcome.